Hello friends, today we will see what are protein structure database. One of the example we'll be discussing is PDB. Myself Febi Paiva, Assistant Professor in the Department of Zoology, St. Joseph's College for Women, Alappuzha. Protein structure database. What are they? As the name indicates, this is a collection of something. Collection of what? The structures, the three-dimensional structures of mostly proteins. So, how does this databases came into existence? It came into existence with the belief that proteins with a common origin, common evolutionary origin, have almost a similar structure, whereas those with dissimilar proteins do have dissimilar structure. Whether it is primary, whether it is secondary, tertiary or quaternary, it may vary in dissimilar proteins. And the protein structures can be predicted with the help of these databases. So what they store? They do have a collection of 3D structures of proteins. And one of the best example of such protein structural database is PDB, that is Protein Data Bank. So here we can see that it is the primary database of protein structure. Primary means once it is directly been archived from the scientist when they do an experiment. From their experiment, if they come up with a structure of any protein, it has been archived there. It has been submitted and archived there. So this is the first hand data, so we call it as primary database. And it is also used to predict the three-dimensional structures of proteins and the nucleic acids. It is believed that it is the one of the single worldwide archive of the structural data, and it is maintained by the RCSB, that is Research Collaboratory for Structural Bioinformatics. Because it is believed that if we understand the shape of a molecule, it helps us to understand how it works. That is, this knowledge can be used to derive the role that particular molecule play. The role of that molecule or the protein in the human health, the role of that protein in causing disease, and the role of that particular protein in drug discovery. So the structure in the archive range from a very tiny proteins, a bit of DNA, to large molecular machines like ribosomes. So it may constitute of tiny DNA structures, it may be having a tiny proteins, structure of tiny proteins, and it may vary up to large molecular sized machines like ribosome. And from where are these data obtained? From where are these structure being retrieved in this database collection? And it has been obtained from the X-ray crystallography experiments done by the scientists, or it can be from the NMR spectroscopy. And once those scientists get the structure of any protein, it has been submitted into the PDB. And the structures may contain atomic coordinates, information about atomic coordinates. It may be having some basic and computed structural details. And also, these PDB collections may have annotations of the depositor. That is, if the depositor have to give some information about any of the coordinates or any of the linkage or any of the interaction, that will be given as annotation, and that is also included in this PDB collections. And the data is in this PDB is freely available to the researchers. And we can see that it can be accessed by the URL http www.pdb.org, or it can be available by simply searching through the Google search engines, or from other search methods also, interfaces also, we can come into this PDB interface. And the format in which the submission is done is PDB. Initially it was only PDB file format. Later on further modifications were done and the files were being submitted or stored in another format, that is MMCIF, that is macromolecular crystallographic information file format, that is macromolecular, C crystallographic information I, and file F format. And nowadays it is better being represented in another version, that is XML version of MMCIF, that is PDBML, and that format of file we call it as PDBML. Now we'll just go with we'll just have a search. We'll just see how to go with this PDB search. Simply we could see that if we write as PDB in search engine, Google search engine, that will take you to the page RCSB, and hopefully you may remember that is RCSB. And if we go with the home page, this interface will be shown, and we could see that here itself they have written about the a kind of introduction about PDB. Now if you want to search any of the protein, like for example hemoglobin, just imagine hemoglobin. You want to search hemoglobin structure. Here comes the search results. That you will able to see that there are so many researchers been, and about 20 there are many research results being shown. And here we could see that organisms, the method how which the structure was being taken, that is through X-ray crystallography, and when it was being released. Okay, so all these ideas will be given. Like for example, if you take another protein, urease enzyme, you could see again so many results, those proteins which are being submitted to PDB. You could see different and like organisms also has been given, some microorganisms name has been given. So this urease belongs to that particular microorganisms, and how and when it was released, and the citation also, who all did the research work here, and everything is been given. And if you want to find it in a Homo sapiens, we'll just see urease in Homo sapiens. Okay, we'll go with another enzyme like hexokinase. So you could see here Homo sapiens. Since we have written Homo sapiens, it gives the results of Homo sapiens. And what we could see is that if I click on this, we could see the structural details of this, and three-dimensional view of this protein is possible. So now if I go with this structure, it will take some time for uploading. Yes, so here you could see the structure, And if you want to see in another format, like you can see in different form, like if you go for NGL, the structural format will be of different way. Now you could rotate the structure. Thing is, you could zoom it also. Can you see this can be rotated? You can zoom by just clicking the structure. You can spin it. You can have it in a full screen structure. You can also go for screenshot. If you want to have in a different format also, you can have it, space fill format. Like if you go for element by element, if you want to screen, you can screen that by element. If you want to see it in a different style, like you want to see in different style, you could see the structure in different style. If you want to see the water molecules, you could see the water molecules. So in this way, we could visualize the structures of the protein, and by using this we can predict the problems, or we can predict the function of the structure. So that is the use if we go in detail. So that's all for the